Hey guys, it's me again, and I'm here with video 12, which is DNA to a protein. So how do we go from the DNA inside a cell to being able to make a protein at the ribosome? Well, here's some things you need to remember. First of all, DNA is translated into amino acids. Remember, amino acids make proteins, and since they make proteins, you've got to also remember that DNA basically codes for proteins. So now let's look at this a little bit. Remember a few things about DNA. DNA cannot leave the nucleus. Ribosomes make the protein and are found outside the nucleus. So now if I just told you DNA codes for how to make proteins, and proteins are made by ribosomes outside the nucleus, and DNA can't leave the nucleus, well, you got to be thinking. There's got to be some way to get this message out of the nucleus. So how does it occur? It occurs through a thing called RNA, or ribose nucleic acid. There's some slight difference between RNA and DNA, but I think about DNA as being the master copy, or the master, I should say, and RNA being the copy. So the master can't leave where it's at, it's found inside the nucleus, but the copy can leave the nucleus. Now, for the test, I'm definitely going to ask you to give me 3 differences between DNA and RNA. So you need to know these for the test. And here they are. DNA has thymine, RNA has uracil. DNA is double-stranded, RNA is single-stranded. And DNA has deoxyribose, RNA has ribose. Now, if I can explain a little bit, you'll notice, all right, over here, this is the RNA side here, and this is the DNA side. The DNA is double-stranded, the RNA is single-stranded. That makes sense by just looking at it. DNA has deoxyribose, which is the D, right? RNA has ribose, which is the R. That makes sense. Now the thymine, if you remember, in DNA, C's always went with G's, T's always went with A's. I think everybody's got that. Well, RNA, C's still go with G's, but now A's go with T's, so you have a substitution. So something, either double-stranded from single-stranded, the thymine, the uracil, or the ribose, the sugar, one of these 3 things allows RNA to leave the nucleus where DNA could not. Now, when you look at DNA, there are 20 amino acids in all in the human body, or in any living thing for that matter, and it takes 3 nitrogen bases to code for an amino acid. And these 3 nitrogen bases are what we call a codon. So over here on the right-hand side, if you'll notice, here's a part of a DNA molecule that has been transcribed into what we call an RNA molecule. And it is G, C, U are 3 nitrogen bases, A, C, G. So that's a codon, a codon, a codon, a codon. It takes 3 nitrogen bases in a row to equal a codon. That's going to be important on the next page. All right. We have a genetic code that is universal. That means for a cat, a dog, an oak tree, a human being, no matter what it is, this code is true. You can look up my RNA, you can find a codon sequence, and you can look it up on this chart, and you can tell what amino acid it codes for. You can do the same thing for an oak tree, a kitty cat, or whatever. So let me show you how this works. If I ask you to look up, let's say, UCA, well, if you'll notice, the first base in the codon is U, so that tells me that that answer has to be out of these 16 possibilities. So these can't be the answer. The next letter is C. C, so it tells me it has to be one of these 4. So these can't be the answer. And then A. It actually tells me that serine is the amino acid that stands for UCA. All right, let's look up CCC. CCC would be this C, and this C. If you look at where they all cross, is that proline right there. So CCC stands for proline. ACC, if you look at it, it's going to talk about this A, this C, and this C. If you look at where they all cross, it's got threonine is the answer there. I hope you'll be able to do this, the genetic code. We'll have lots of practice so far as with codon bingo in class. There's a chart in your book, if you'll look at it. It's a lot easier than this chart to look up the amino acids. You actually start from the center and you work your way out, and that's the one we'll use whenever we do codon bingo. Now, before I go any further off this page, don't you notice the top up here? this structure right here is called a tRNA. Now, a tRNA is called a transfer RNA because it brings the amino acid to the ribosome. That's what this is right here. That's the amino acid. Okay, so this tRNA here brings the amino acids to the ribosomes where they can be assembled. All right, now this chart here is just a blown up version so you can look at it. So if I asked you to look up, let's say AAA, if you looked it up, hopefully you would have looked up lysine. What if I asked you to look up CAU? CAU, hopefully you would look up histamine. Now I want you to notice something. There's 3 lines here. These stand for stop. Okay, these tell the ribosome to stop reading the link. And then there's one here called methionine. AUG is this codon. Methionine is what we call the start codon. So these two or start and stop, are also included into the genetic code. All right, now there are 3 types of RNA: mRNA, rRNA, and tRNA. mRNA is called messenger RNA. mRNA is the RNA that's made from the DNA that carries the message from the nucleus to the ribosome. rRNA is ribosomal RNA. It's used to code for ribosomes. Makes sense. And tRNA is the one I showed you just a minute ago. tRNA's purpose is transfer RNA, and it carries the amino acid to the ribosome where it can be assembled. Know these 3 for the test. All right, the next thing here is there's two major processes dealing with DNA and RNA. They're transcription and translation. Transcription results in the formation of mRNA from DNA. Basically changing DNA to RNA is transcription. And if I were to transcribe something, I would simply copy down somebody else's words. That's basically what's happening here. DNA is being copied into mRNA. Basically the same information is being copied, just in a different format. And then the next process is translation. That's when something is actually taking the mRNA and changing it to an amino acid sequence, which eventually becomes proteins. Now, if you think about translation in the classroom, if I was signing in front of the classroom, there would be someone translating what I was saying so that you can understand it. And we'll do a little activity in the class with this. You can also look up this activity on Edmodo and look in protein synthesis, and there's a little activity that goes through transcription and translation. Really good one. All right, and here's 2 processes basically defined. If you'll notice, transcription is up here at the top. Here's your DNA, and it's being changed into mRNA. And notice where that's happening at. It's happening in the nucleus. Then that mRNA leaves the nucleus, and when it leaves the nucleus, that mRNA is being read by a ribosome. And this funny looking structure here is a tRNA. A tRNA is bringing amino acids to the ribosome, and then those amino acids are being put together into what we call a protein. So that is translation. Transcription happens in the nucleus. Translation happens outside the nucleus. Okay, guys, I hope video 12 is helpful to you. And if you have any questions, please let me know.